Good afternoon and welcome to the POOLBEG PHARMA PLC investor presentation. Throughout this recorded presentation, investors will be in listen-only mode. Questions are encouraged and can be submitted at any time by the Q&A tab situated in the right corner of your screen. Just simply type in your questions and press send. The company may not be in a position to answer every question it receives during the meeting itself. However, the company will review all questions submitted today and publish responses where it is appropriate to do so. Before we begin, I'd like to submit the following poll. I'd now like to hand you over to CEO Jeremy Skillington. Good afternoon, sir.
Thank you, and I appreciate the introduction. Thank you, everybody, for joining us this afternoon, this Monday afternoon. Today we have some key opinion leaders, experts, to discuss POLB 001 and our direction into oncology. I think right now we've talked quite a bit on the role of POLB 001 in severe influenza, and earlier this year we filed some patents for the use of P38 MAP kinase inhibitors in this kind of cytokine release syndrome, in this oncology setting. So we want to give a bit more background to it, and so we'll go through the presentation. What we do have a nice agenda. We've got 90 minutes for us to discuss. We can be flexible on time, but high level is I'll give an introduction to POOLBEG and the opportunity that is now 001 in cancer immunotherapy. I will pass across to one of our key opinion leaders, Dr. Martin Kaiser, to discuss. Again, I like to think this is really at the cold face. This is people at the front lines in the medical community to how these drugs, cancer immunotherapies, are used, and some of the issues, the side effects that we believe 001 can address. And then I'm very excited myself personally to hear a patient's perspective. So we're fortunate to have Bob Monroe join us. Bob's going through treatments for many years around multiple myeloma. He's going to tell us about his journey, about his challenges, and also some of the issues and side effects that he had, again, where we think 001 can play a role. And then finally, we'll wrap up with our good friend and colleague, Professor Brendan Buckley, who sits on our scientific advisory board as well as our board as a whole, and talk about 001 and the role that it can play in cancer immunotherapy and acute inflammatory conditions. And then we'll have a general conclusion and wrap up. So again, really glad you could join us this afternoon, and for us to go through this story. And it's quite a fascinating and exciting story for POOLBEG as we move this forward. Again, as I said, the biographies, Martin will introduce himself, but he's affiliated with the Royal Marsden Hospital, obviously focused in hematology space, and seeing patients for many years. He'll tell us some of his insights into the space of cancer immunotherapy. Bob, that I mentioned, myeloma patient advocate, for a fantastic story to learn from, and of course Brendan, who's he's been involved in POOLBEG from the start and provides a lot of expert guidance as we move through the clinic with our programs. So as an introduction, just to ground you all, many of you may be familiar with the POOLBEG story, and some of you may be new. So I just want to take a few slides to give some context to our plans or our discussions today, and in particular the potential for 001 in cancer immunotherapy. So as a background, we're developing innovative medicines to address unmet needs. We did start in the infectious disease space as we spun out of hVIVO, but now we're getting ambitious. We're branching into other areas where there's clear unmet needs, and we see that POOLBEG can play a significant role. So we have an exciting pipeline of innovative products. So we have, and again, talked this year, we've been incredibly productive this year. We have 2 collaborations around the artificial intelligence mining of clinical trial data, infectious disease clinical trial data in influenza and RSV that we spoke to this year. We also are near the clinic to begin studying an oral GLP-1 program. Again, that's for diabetes and obesity and beyond, getting that into the clinic next year. And again, the purpose of today, we'll talk about POLB 001 with broad applicability into acute inflammatory conditions. So very excited about the direction that molecule is taking since we got our clinical data early this year. As a biotech company, we see ourselves having a unique model where we take these programs, move them forward into the clinic, get early human clinical data, that's safety and efficacy, and then partner with pharma. So that's always part of the goal. Let's get there to have that key data so we can partner with pharma. And as you see, we're kind of driving towards that and feel that 001 in immuno-oncology adds significant interest and value to the program. And again, committed to doing deals early. And I think that links very well with our other point about the exceptional team that we have here, as long with our exceptional track record. So again, we've generated over $2 billion in shareholder value for various companies that we've worked with in the past. Many of you know Cathal Friel was involved as a co-founder to Amryt Pharma back in 2015, who then were acquired earlier this year by Chiesi Pharma for $1.48 billion. So again, significant return or significant success in driving share company value. And then myself was involved recently in the acquisition of Infosome by Roche. And that was a company that was founded in 2016 and exited in 2020, 450 million upfront with downstream milestones that we hope to come. And again, Poolbeg as a company, we have a strong financial position. As I say, we have a capital efficient operating model, had a cash balance of 14.1 million in the middle of this year. And again, adding to that, we have non-dilutive funding to help bring our programs forward. So we've been successful in that regard. So, we're happy with the direction it's taken. Again, just a snapshot of the pipeline as it stands today. 001 we mentioned. I mean, this is our lead asset. Very excited about the potential now in these acute inflammatory conditions. We're progressing this forward as phase 2 ready. We had a very exciting LPS challenge study outputs from earlier this year. Brendan will talk to that later on. And beyond the infectious diseases now moving into oncology, and obviously the purpose or topic of today's discussion. So again, phase 2 ready, and discussing with our pharma partner colleagues about collaborating on that, driving that forward to get key data around cytokine release syndrome, which is a significant issue for these newer infectious or newer immuno-oncology treatments. Again, I won't talk about the rest of them, but again, very active programs and very excited where the programs are progressing. I think what's exciting about this is kind of on the back of filing intellectual property early this year about the use of p38 MAP kinase inhibitors in oncology, and generating the data from our LPS challenge study. We've had several discussions with hematologists, and we were delighted to have a poster presentation accepted for the upcoming American Society of Hematology annual meeting in December in San Diego. And this will be driven by our collaborator, Emma Searle, who'll be presenting based in Manchester. But again, we see the potential of POLB 001 to play a role in cytokine release syndrome, in addressing an issue that, again, a lot of hematologists have issues with these days with the new treatments. So again, the ASH conference in San Diego, that's 25,000 of your hematology experts, your scientists, your clinicians, coming in to learn about the newest takes, the newest update, the newest science around hematology. So we're delighted to be able to have a poster presented at that conference to maybe introduce some to Poolbeg, but obviously discuss with our pharma partners about the role that benefits the POLB 001 can play in addressing CRS. So again, we feel that it holds great promise in tackling CRS, leading to improved patient outcomes, reducing, I think it's important, to strain on the healthcare system as well. And Martin will talk to that later on. And while we're making these new novel cancer immunotherapies more accessible to a broader patient population. So part of the team is heading to San Diego in December to be part of those discussions. So very excited with the positive reception that we had for that. And again, to ground people, just to help you understand the direction that we're coming from, and the issue around about cytokine release syndrome. As a reminder, POLB 001 is a potent selective p38 MAP kinase inhibitor. This is a key protein involved in the immune or inflammatory response. And what happens in a cytokine release syndrome cycle, and I think many of us are familiar from the COVID-19 pandemic about the issues around severe effects or severe disease. So what you typically get is immune cell activation. That's a normal process. The immune cells then secrete these cytokines, which recruit more immune cells. And in a normal setting, then you come resolve, everything calms down again, and everything goes back to normal. But you have this issue with some patients, a subset of patients, where you have this continual cycle where the immune cells are further recruited, more cytokines are secreted, causing more immune cells to be recruited. And ultimately what you get is this cytokine storm, this hypercytokinemia, which drives the breakdown of tissues and organs, and you see organ damage, and ultimately can lead to severe disease and possibly even death. So we believe that POLB 001 is a potent and selective inhibitor of p38 MAP kinase, can block this cycle, can block the cycle of these immune cell activations, and then ultimately allowing the immune system to calm itself back down, to bring back down. So we see great potential here. And as I say, we started in the severe influenza setting, but now moving more into the cancer immunotherapy setting as well, because we see roles to play here. And this is explained on the right-hand side, as I say, the triggers of these CRS, the drivers of cytokine release syndrome, whether it is infectious diseases like influenza or COVID-19. And indeed, more recently, we believe in cell therapies and immunotherapies, that there's a window of opportunity, for POOLBEG to go in that direction, to generate key clinical data in that direction, but ultimately then, assisting and helping patients and benefiting the healthcare system as well. So again, we see, terrific opportunity in this space as well. And again, as a company, as I said at the outset, we're very much focused on partnering. We don't intend to bring these programs all the way forward. It costs a lot of money to bring it into our phase 2, phase 3 and beyond. But we know that a lot of pharma companies are looking for opportunities like 001 to bring into their pipelines, to help them move their programs forward. And we've had some terrific discussions with pharma partners in the cancer immunotherapy space who see CRS as an issue. So from that, they see that we have complementarity, that we can get our drugs combined together and benefiting patients. So obviously there's a clear unmet need, and as Martin would speak to, it's becoming more and more prevalent as these immunotherapies or more of these immunotherapies are coming online. So as I say, we see strong interest, some fantastic discussions about looking for these better solutions. And again, they see from a pharma partner's perspective, if we can resolve the CRS issue, they see there'll be more appetite, putting patients on these drugs. They spend less time in hospital, which is benefit to the healthcare system. But because of that, there's more beds freed up for more patients to get their drugs. So of course, pharma are interested if we can resolve this CRS issue. So we're very, as I say, excited to be moving forward and testing this clinically is the plan for next year. So again, there's different types of deals. We know that pharma would provide drug for this application, but then from there beyond, it's funding the clinical trial, for example, or even doing options and license deals where we can generate revenue from that. As I say, we're a deal-making company, so that's part of the plan or the strategy when it comes to our partnering around POLB 001 in oncology. And again, very excited to test it in an initial clinical trial, but then we see broader applicability beyond that in the oncology setting, as I say, as these newer immuno-oncology drugs come online. And as you'll see, again, Martin will present, there's very exciting pipelines of products coming through. So again, we can see that POLB 001 can play a role in many of these to address or alleviate the cytokine release syndrome, the CRS issues that they have. So that's just a general introduction. As I say, I'll pass across now. I'm delighted to have Martin Kaiser on board with us today, again, to provide his knowledge and his expertise. And as I say, from the coalface, this is kind of, a physician, a doc, seeing patients day by day and talking about the, in the myeloma space. So Martin, welcome, and looking forward to hearing you, share your thoughts and insights.
Thank you, Jeremy. And yeah, very happy to provide really a direct experience with CRS and what that means for us practically. So I am here and I'm a consultant hematologist at the Royal Marsden Hospital, as has already been mentioned. I'm also a translational researcher. I have quite a bit of experience in this role, and I specialize in the treatment of a disease called multiple myeloma, which some of you will be familiar with, as there has been a lot of progress, especially in terms of new drugs over the last decade or so. And as part of that, I have been playing a role as a principal investigator, also chief investigator on some academic trials from early phase, first in human to late stage clinical trials. I'm also a vice chair of the academic UK Myeloma Clinical Trials Group, the UK MRA, and I have some role in international policy discussions through roles with the European Hematology Association and the European Medicines Agency's Cancer Medicines Forum, for example. So I have an interest in generally, the progress that has been made, but particularly, of course, in more recent years in terms of the immunotherapies that have been really transforming how we have been treating patients. And I think what we have seen probably a little bit earlier to start already in hematological malignancies, of course, in solid malignancies has really reached now a very, very broad level and reaching larger parts of the oncology population. As you see here on the left-hand side, the typical chemotherapy survival curves in the past all unfortunately reached more or less the x-axis over time, mostly because these treatments could only be given for a certain amount of time. Otherwise, they caused side effects that were equally leading, unfortunately, to severe issues for the patient. And then with the introduction, of course, of PD-1 inhibitors in solid oncology particularly, we have been seeing this gradual increase. It very much reminds of how combination chemotherapy in the decades before I started practicing suddenly led to survival curves showing some long-term survival in hematological malignancies. Now the same seems to be happening with the immunotherapies across the board in oncology. And I think that is really the big promise that we're seeing, and it's no wonder that that has led to a massive uptake in their use. I think if we talk about immunotherapies, of course, we can mean so many different things, and many of you will be aware of the many different approaches that have been tried out and explored over the several decades, in which it was always clear that the immune system can play a huge role as a partner in ultimately targeting the tumor. Probably not all of them have been equally efficacious, that you can see listed here on this slide, but clearly the immune checkpoint inhibitors on the top. And then probably treatments that are more related to what we see at the bottom here have more recently shown the biggest promise and the biggest steps forward. And I think that's what I really see in my practice very clearly being now a day-to-day routine, is unlocking the antigen-specific T-cell responses, and that can be either through unlocking the responses that a T-cell would probably already have on its own in immunologically hot tumors. But more recently, what is really exciting, especially in the field of multiple myeloma that I work in, is that we can give these T-cells that normally don't really recognize myeloma cells and many other tumors, give eyes through different mechanisms. And on the right, lower right-hand side, you see here where before we had only naked antibodies, we can now modify effectively the T cells directly in terms of guiding them towards the target, towards the tumor, either in terms of genetically modifying them to express receptors as in CAR T cells that would tackle the tumor, or in terms of infusing monoclonal antibodies that are effectively building a glue between the T cell engager, between the T cell and the tumor, in form of T cell engagers, or also bispecific antibodies, as we call them. And importantly, of course, the big difference that this has made is the specificity of the treatment, the low toxicity to healthy tissues. Actually, what we're seeing in myeloma at the moment is an unprecedented durability of the responses to single agent drugs that we have in that shape, I think, not seen with a new treatment before. And of course, there is, at least in some diseases, even evidence turning out that we might have even an immunological memory or immunological response that can last decades. Not in all treatments yet, but there have been recent papers, for example, in the related disease chronic lymphocytic leukemia, where CAR T cells were alive still many years after their initial infusion and were still working against the tumor. So this is all very, very exciting, and it really is, from a physician perspective, a great time to be active in this field. Now, not surprisingly then, I think the projected use of these drugs will only increase over time. There is, from my perspective, as using these drugs on a day-to-day basis, no doubt about that. What we have seen before with monoclonal antibodies that you see here on the left-hand side, for example, the hematology rituximab, but of course many later in solid cancers as well, will very, very likely and is already being replicated in terms of our use of these new T cell and cell and gene therapies effectively, because of their efficacy, but also because of their really lack of classical toxicities that have in the past really hampered the long-term use or the most effective use of these treatments. So, going a little bit into detail in a drug that I'm using, for example, on a daily basis, bispecific antibodies in terms of teclistamab here, which is a BCMA targeting bispecific antibody for multiple myeloma. This is data from a trial in relapsed refractory patients, a really early phase trial, where this drug was given as a monotherapy, and that's quite unusual. Myeloma was one of these first treatments, these first diseases where combination therapies were becoming more and more common, because they were needed, at least in relapsed refractory disease, to still get response rates above 30%. And here we see with a single agent drug that the response rate was over 60%, which is really unprecedented. And actually, as you can see on the left-hand side, the median duration of response is really striking. We see many patients that are still having ongoing undetectable disease several years into the treatment now. But on the right-hand side, you see what is clearly happening with this treatment, what we have not seen with, for example, many of the naked antibodies that we used before, is that we see cytokine release syndrome, which has been mentioned before already, as being an overreaction of the immune system and an overexcitement in terms of cytokines being released. which I will explain a little bit more over the next slides. Now, most of them, fortunately, are relatively low grade with this drug, but as you can see, it cannot be ignored if 70 or more % of patients that get the drug are getting cytokine release syndrome. Now, what does cytokine release syndrome really mean? I will go through the specific side effects in the coming slides, but overall, it's really as if the patient had an infection and is really having fever and similar sort of symptoms or more. These can really be quite substantial. Now, what that practically means for us is most of these treatments we have to start as an inpatient for safety reasons, either because we have to ramp up the dose over, let's say, the course of a week. That's normally the case in bispecific antibodies, for example, that we use in myeloma, but also some of the CAR T-cell therapies in theory would not require necessarily a much longer stay if they did not cause any issues, because the procedure as such is not particularly demanding an inpatient stay. So on the very top is patient 1, you would see actually practically we do know that we need to admit a patient at the moment, at least with the standards that we have at the moment in terms of CRS management, for a relatively short-term stay, so that we can actually monitor cytokine release syndrome and, if need be, treat them. And if the patient does not develop any of these, they can actually be discharged after a 7-day stay. And that's actually allowing, of course, in a system where often there is a shortage in bed time, a clear infrastructure planning. That's when the patient goes home and I can admit the next patient to start this treatment. Now, the example here of patient 2 is showing what happens, however, if the patient develops a mild CRS, and that is sometimes more tricky than one thinks. This often doesn't need very sophisticated treatment. One often just waits with help of some fever lowering drugs like paracetamol, that the CRS settles on itself again, but it often delays when we can give the next dose. So suddenly the stay, inpatient stay that before was actually quite calculable has now become more incalculable, and there are several days where the patient just waits for the fever to settle down. So suddenly we extend a 7-day inpatient stay to a 10-day inpatient stay. And that can, of course, be far more serious, often more likely with CAR T-cell therapy. We don't see that normally with our bispecifics, at least, but I think with all the treatments that come along, it has to be seen for every drug individually as to how severe the CRS can be, because in CAR T-cell therapies, we often have to have a intensive care unit in the backhand. And if the patient really develops severe CRS, despite the treatments that we can offer currently for CRS, they might have to be admitted to the intensive care unit for a certain number of days. And suddenly a stay that is quite uncomplicated for 7 days can be extended, to a multiple week stay, with, of course, associated negative experiences for the patients as well, if they have to be on a high dependency unit. So how can CRS effectively present itself? Well, I mentioned already the most common way how it presents is a way of fever or something that would mimic an infection, whereby the patients, through the cytokines that are floating through the body, through the immune system that is activated as if it was an infection, feels fever, a raised heart rate. What can make the situation more serious is the shortness of breath that we see, or a drop in oxygen levels where the patient might need oxygen supply, and a lowering of the blood pressure, which can really become quite serious. And if that is more serious, the patient might need monitoring on a high intensity unit. Now, if we talk about cytokine release syndrome, we also have to mention ICANS, which is actually immune effector cell associated neurotoxicity. So effectively, mostly symptoms caused by the brain being affected through the immunotherapy. And there is still an ongoing need to understand how much of this is really caused primarily by the same mediators that are causing the CRS, and how much of it can be specifically caused by the treatment or by other factors. They can happen either together, which is the more common one, CRS and ICANS, or they can happen also slightly timely delayed or completely independently. So ICANS is something that can develop into a very serious condition for the patient as well, with confusion, and even seizures happening and often needing intensive care observation of the patient as well. There are some additional adverse events that are just common and often caused by the treatments that we have to give for the cytokine release syndrome, as well as by the cytokine release syndrome itself. And that is actually particularly a lowering in blood counts, white cell blood counts, as well as platelet counts, often then related to that an infection risk. But sometimes even more severe cytopenias and more general side effects that we can see. So these are all different for each immunotherapy drug that we're using. And it's quite interesting, of course, with ESMO just having happened to see that with immunotherapies now taking a foothold increasingly in solid oncology as well, that many of these are overlapping, are really a common feature across immunotherapies for hematological oncology or solid oncology as well. And the individual side effect profile and severity will always be have to be determined for the individual drug. You can see here the treatments are only increasing, and many of these treatments here, be it the CAR T cell therapies that have received marketing authorization, or the bispecific antibodies, of which several are actually for the disease I'm treating, multiple myeloma, have received marketing authorization in the last years, many even just this year in 2023, but they all require CRS management, and we have drugs at the moment available for this. The key drugs that we're using for CRS management are tocilizumab, dexamethasone, and anakinra. Dexamethasone is probably the most unspecific drug and probably also the one that has the most side effects, both short-term, also longer-term in terms of immune suppression. The other two drugs are monoclonal antibodies that are effectively acting against elements of the cytokine storm that is being the underlying cause for the cytokine release syndrome. However, they all come with their individual ups and downs, and I think there is clearly room to improve on the options that we have available for CRS management. And if this be alone for the case that at least some of these drugs have to be given as an IV treatment and thereby require the specialty environment of a hospital and an inpatient stay for the patient to manage CRS. So what we can say as well is that, of course, there has already been very innovative attempts at trying to see whether we can use the already available drugs maybe in a more preventative fashion. So this is data from, again, a drug that is currently under investigation, doesn't have a license yet. It's called cevostamab. This is a drug which probably has a bit higher and higher grade CRS than some of the other drugs that we have currently in use and licensed already. And it was quite obvious that this would be a prime target to try whether the prophylactic use of tocilizumab before giving the first dose of this treatment would completely abrogate potentially the CRS risk. And although it clearly lowers the risk, as you can see here on the left-hand side, from 90% to about 40%, it does not abrogate the risk of cytokine release syndrome completely. And that ultimately doesn't really then mean that there is a real impact on the management of these patients when they start the treatment. So because there aren't clear predictors who these 40% are, it would still mean that all patients would have to be admitted because they might need a second dose or even another treatment to treat the cytokine release syndrome. Now, very reassuringly, on the right-hand side, however, you can see that despite giving prophylaxis, there is no negative effect on the efficacy of this drug. It might even look as if the response rate was slightly higher in these that received tocilizumab prophylaxis. Whether that's a true signal, I think that would need definitely more patients, and I think is debatable. But what is quite clear is that there is no detrimental effect of being more proactive generally around cytokine release syndrome, and that not being an essential part of the efficacy of these treatments. So this slide is actually here to show a little bit how heterogeneous the different treatments are that we're giving in terms of onset, duration, and difficulty of resolving actually the cytokine release syndrome. So on the top of this slide, you're seeing some typical CAR T cell therapy data. And the blue line here shows the time to the onset of the cytokine release syndrome, which can really be up to 2-3 weeks, sometimes after the drug was given, probably to do with that CAR T cells grow inside the patient's body and thereby can still later on lead to cytokine release syndrome. and likewise the duration of the cytokine release syndrome being longer in these treatments, whereas the typical bispecific antibody therapy here, the myeloma drug, teclistamab, that I've already shown, normally has a quite early onset that also doesn't last as long, and might in the future then therefore need other strategies in terms of cytokine release syndrome management. But as you can see from both of these, there's clearly room to improvement for management options. And that actually brings me to my final slide. So we have really a number of challenges. As much as we have a promise and a clear increase in use of these new treatments that redirect or reuse T cells to address attack the tumors, there is clearly a rapidly growing number, and there is an increasing level of adverse side effects. We need to understand really individually as to what these treatments do. But I think there is clearly an increasing development of off-the-shelf therapies, cell therapies, but also bispecific antibodies that will have very common features in terms of cytokine release syndrome across hematological oncology and solid oncology. There is a complex administration management of the cytokine release syndrome drugs that we have at the moment, and they are posing a burden on the healthcare infrastructure as in terms of giving these drugs in the first place. So the pure fact that I have to start every patient as an inpatient at the moment who I start on one of these new drugs, of course, is a massive impact given the prevalence of myeloma in this situation on the system. And there are better treatment modalities really needed that would facilitate this to manage the toxicities of cytokine release syndrome. But of course, there's also going to be a growing number of patients living with these conditions with improving overall survival, which will yet create new challenges for follow-up. And I think reducing patients' need for inpatient stays is one of the key improvements that we can make over time, even in diseases that now become more and more treatable. I think there's still a lot of headway to improve the patient experience and the utilization of healthcare resources. So thank you very much for your attention.
That's great, Martin. Really appreciate that overview, and I think it really, striking or highlights the advancements have been rapid. They've been terrific. I think utilizing the immune system has been around for a while, back in Coley days and TLRs, but right now it's a case of moving forward in leaps and bounds. But these issues, the CRS issues, need to be addressed and adapted. And as I say, we're excited at POOLBEG to be able to kind of have a role to play in that space. So again, appreciate the overview. Of course, we'll take questions from Martin at the very end. But the next step now, I think I want to introduce Bob Monroe. And Bob, again, I use the word again, this cold face. Bob is in the middle of this. He's as obviously he'd been through myeloma treatments over many years, and it's a fascinating story. And Bob, I won't say much more. I'll leave it to you to give the full story. But really looking forward, and thank you again for joining us.
You're welcome. Well, good afternoon, everyone. As Martin's introduced, working at the coalface, I guess I've got the coalface within me. So I'm going to tell you a bit about my perspective on how it looks. I'm just going to talk about why I'm actually an atypical myeloma patient. My qualifications for speaking to you today, my journey, my drugs, what they've done for me, and what they've done to me, which is quite significant, and really talk about the side effects of these drugs, which is what we're talking about today, and the quality of life it results in for patients, and potentially what this drug could do for us as patients, and as we've heard, for the health services as well. So atypical patient, well, the reason is myeloma is a very, very heterogeneous cancer, meaning it's different for absolutely everybody, and it's even different within patients. It morphs into different things over the years, why it's so hard to treat and why we haven't come up with a cure yet. The drugs that have been found, which I'll talk about, have been incredible, but they've generally only lasted people for a year or two or less, sometimes a bit more, 5, 6. But eventually they stop working. The cancer finds a way to overcome the drugs. You need another one. So atypical, yes, I'm a typical atypical patient. We're all completely different. My qualifications for speaking to you today, I've been treated for 12 years. I was diagnosed in 2012. I've experienced most of the existing drug types and experienced most of the side effects. And all those I haven't experienced, I picked up second or third hand from being a Myeloma UK patient advocate. So I've spoken to literally hundreds of patients. I just should as a disclaimer, really, I'm a bit cup half full type of guy, and my pragmatic optimism gives me a lot of strength. So you won't get a sort of a tale of woe from me, but nonetheless, I think it's useful for today's process. So just a quick background on my treatment. 2012, as I said, very lucky diagnosis. Went skiing with my mates, got pneumonia, rushed back into emergency hospital back here, and found that I'd got myeloma, which is not the best news I've ever had, especially when the consultant told me it was incurable, and he specifically said, "Don't Google it." I thought, "Oh my God, that doesn't sound too good." And the reason it doesn't sound too good is because when you do Google it, which of course I did, was the survival statistics at the time. I think there are about 20% of people weren't lasting 2 years. So on average, 5-year survival was only 45% of patients, and only 19% of patients lasted for 10 years. So that wasn't a good start. I was lucky, though, as I said, lucky diagnosis, because I went skiing and it was caught in A&E, which often happens to patients. They end up with a broken leg or a rib or something like that. They get rushed in hospital and they find out they've got this underlying myeloma condition. I didn't have any catastrophic bone damage or catastrophic kidney damage. So it was a good start for me to pick up the disease quite quickly, which become relevant later on. So the gold standard treatment for myeloma then, and still is, a stem cell transplant, a transplant of your own stem cells. They take them out of you, kill you with a heavy dose of chemotherapy, and then reimplant them. And the idea of regrowing your immune system in a more in a cleaner state, if you like. But before you have that, you have a couple of these novel drugs that they've been developed over the last 20 years. In my case, I got thalidomide and Velcade. Thalidomide, as I'm sure you're aware, very notorious drug, actually offered to my mother back in 1959. It was for morning sickness. Fortunately, she didn't take it. So it was obviously a banned drug for a long time, but then it was repurposed as for leprosy, I believe, and then eventually for myeloma. And interesting to note that I understand that the reason our regulatory system is so good in this country in terms of looking at drugs and looking at their safety, etc., was this terrible incident with thalidomide, and so it has a good legacy in some ways. Anyway, it's repurposed for myeloma. It didn't work for me. The Velcade didn't work for me. So after the first year, things were looking pretty grim already. Your class as refractory and to drugs, and it was looking pretty grim. But along came another novel drug, Revlimid. And as you can see, the whole list of drugs there: Revlimid, pomalidomide, the first monoclonal antibody for myeloma, daratumumab, carfilzomib, ixazomib, elotuzumab. I've had the lot, full house. You can see from this chart here, which is obviously not linear. I'm indebted to Martin for this. Rhubarb and orange peel. What was that all about? Back in 1840. But you can see all the drugs have really been developed in the last 20 years. All the ones in orange, all the ones I've had. So quite a lot of them, 8 or 9 of the drugs, and also in various combinations, which, as Martin alluded to, is how the myeloma drugs seem to work best of all, producing much higher response rates. But in all cases, the drugs in my case only got me down to what was called a plateau level. So basically lower levels of the existing biomarkers. That's my phone just gone off. And here we can see what we were talking about in terms of the survival rates. This is survival rates. You can see the lower red line is probably what I was looking at back in 2012. Where if you look about 10 years looks at about 19% survival rate. And the latest projection, bear in mind these figures are also obviously lagged because you need time to survive. But the yellow line shows that we've actually going to see probably a 4-fold increase in survival rates, certainly for 10 years, and commensurate rates over the rest of the curve. So quite remarkable. And I've been very, very fortunate to get myeloma when I did, rather than say 15 or 20 years earlier. Now that took me up to about 2 years ago. But by 2022, I'd actually used all of those licensed drugs that were available at the time. And fair to say, I've had a death rattle, a literal death rattle, because my bones were breaking. They became increasingly brittle. My vertebra had collapsed once. I had about 8 rib fractures, sometimes for the most minor reasons, a cough, rolling over in bed, a wave hit me when I was in Cornwall. And I'd seen this before with other patients, and I knew that's probably the worst sign possible. It's getting close to the end of the road. There didn't seem to be much else to take in terms of drugs. I've been following the developments in CAR T-cell therapy, and over the previous 2 years leading up to 2022, there was much promise that they would be available quite soon in the UK. But it kept getting put back. There's lots of manufacturing issues there. And in fact, they're still not available in the UK today because of these manufacturing issues. More prevalent in the States, starting to come in Europe, but not in the UK. But in the meantime, they developed another product, bispecific T-cell engagers, which operate in a similar way, but are not quite so intense in terms of, well, manufacturing. They're basically off-the-shelf type CAR T-cell engagers. I'd also followed that very closely. I'm a complete nerd when it comes to myeloma drugs, for a good reason, of course. So I followed these bites, as they were called for short, and was told that by a couple of hospitals that actually this would be a great bridging therapy. And in February 2022, I managed to get on one of these trials for teclistamab, the same one that Martin was talking about, on the second phase of this Majestic 2 trial, which basically added another existing drug to teclistamab. And Martin said that the mono teclistamab trial produced results of 60% response rate, which is absolutely remarkable in the myeloma world. Well, it looks like the teclistamab 2, which is teclistamab plus another drug, the response rates are even better. Moreover, and this is the big one, after 12 months, I got my first ever complete remission after 12 years of continuous treatment. A minor miracle. Minor miracle for me, but actually all the patients I know taking this drug and all the anecdotal evidence I have is that the response rate is very good. The complete response rate is off the scale compared to other drugs. And so far, none of the people I know and I know quite a few on this drug, have actually relapsed. So 21 months and counting. So don't mention the C word. I mean, cure. Don't mention it. But, you never know. So, as I said, cup half full kind of guy. I've not wasted my time, my 12 years. I've been doing lots of stuff. This is the happy bit of the story. Saw my son get married, lived my life vicariously through my son playing professional rugby, seen the 2 granddaughters I never thought I'd see, taken up cycling, cycled over Tour de France mountain passes, cycled to Paris 3 or 4 times, raising money for Myeloma UK, and just got back from Italy, where I took 50 friends and family to raise money for Myeloma UK. We cycled from Pisa to Rome. So again, actually, I'm not an atypical patient. I am an atypical patient, I should say, that I do a lot of this stuff, but it's given me a second lease, and I'm very, very grateful, which all sounds fantastic, doesn't it? But here's the bad bit. So side effects, quality of life. So I'm going to rattle through these because there are so many, but major things: I've lost a sight in one eye. I had a cracked sacrum in 2016, collapsed vertebra 2019. As I mentioned, 8 fractured ribs when I looked like I was finished, a fractured arm. I've had heart issues from the drugs, peripheral neuropathy, which I got in the first year, which I still have 12 years later, all sorts of stomach problems. Remember, this is not chemotherapy. This is not the traditional oncology drug where you lose your hair and you're sick. You can be sick. At one point, I did lose my hair through taking a chemotherapy drug for another reason. But actually, you look pretty normal when you're taking this type of drug. But what has it meant? In the 12 years, I think I've had over 500 days in or at hospital. I've had 250 cannulas inserted. You've probably all had a cannula inserted at some point. If you haven't, I wouldn't recommend it. Sometimes it takes 6 attempts to get into my blood, into my veins rather. Another 250 blood tests, 6 bone marrow biopsies. I wouldn't advise anyone to have one of those. At least 30 MRI, CT scans, loads of sessions of radiotherapy, self-administered white cell booster syringes, Red cell booster syringes, that was EPO. I was quite pleased with that one because it's obviously Lance Armstrong's drug of choice. So I was very happy as a cyclist. 30,000 eye drops. And over the years, average about 25 to 30 additional tablets a day. Antiviral, antibacterial, antifungal, anti-shingles, steroids. We mentioned every type of painkiller you could possibly imagine. So over 100,000 tablets in 12 years. Am I bitter about that? Not at all. I'm a big fan of pharma. The myeloma drugs have kept me alive against all the odds. I mean, they really have against all the odds. So I'm a big fan of pharma, and I'm happy to take tablets. What I'm not so happy about is cannulas, time in hospital, etc. But you can see here that it's all these things I've managed to do in my life, which have been fantastic. It's when people say, what job do you do? And I've had a couple of part-time jobs over those 12 years, but not really managed to be well enough to have a full-time job. So my job, staying alive, as the BG said. So let's get down to specifics of bispecifics. As I said, teclistamab was the drug that I was given in February 2020. As Martin mentioned, CRS, viral infections, neurotoxicities. I can't talk about neurotoxicities because I didn't get any. But did I ever get CRS and viral infections, particularly viral infections? Yes, I did. So basically, these drugs, they activate your T cells, but they suppress your B cells in this case. What that means is you get so many more viruses. It's just unbelievable. So if we look at how my initial treatment worked, it was let's see, the first 2-stage doses, I got grade 1 CRS, so mild. I'm very lucky. I got paracetamol for that, but I was in hospital, as Martin said, for 10 days as they monitored me. I then got a virus, CMV, which is a childhood virus, which 50% of us carry, reactivated by this drug because my B cells were suppressed. So a 2-week break, then you have to come back, restart the treatment at the second dose again. Again, grade 1 CRS, paracetamol, very lucky, another 8 days in hospital. Then I got COVID. Then I got some other severe drug-related back issues, some viruses. So I had to restart the second dose again, second and third doses again. Grade 1, slightly worse this time, CRS. So I had this existing CRS antidote, which I'm not even going to try to pronounce. Another 10 days in hospital. So you can see the pattern. There was 28 days in the first 6 weeks, I think, when I was in hospital. I mean, literally an inpatient hospital. It was some of the worst times I've ever gone through because you are in a quite high dependency unit. It's not ICU. There's a lot going on. You can't sleep. There are people around you who are much more distressed and ill than I was. I'm, as I said, cup half full guy. That was the worst moment in the 12 years in terms of treatment. But just being in that hospital, not being able to get out because they were so worried about the CRS coming back, etc. It was truly dreadful. And if we go a little bit further, in the 12 months when I eventually got my complete response, I had this CMV virus coming back all the time. As a result, the treatments were interrupted all the time. I had 10 separate respiratory viral infections, polymyalgia, another drug-induced side effects. So they all interrupted the treatment with teclistamab bispecific. Each required lengthy inpatient and restarts of the CRS protocol. And of course, each hospital stay increases the chance of infection. I think we all got an anecdote about that, about someone going in hospital and picking up an infection, rather ironically. So in that first year, I had a total of 100 days in or at hospital. Dreadful for me and very, very expensive for the health service. Effectively, I'm a bed blocker. I mean, I didn't even feel bad for most of that time. That's the irony of it. So finally, that story just confirms what Martin has said from the patient point of view. The bispecifics, to me, they look like game-changing drugs. I've said this a few times. I said about monoclonal antibodies, but they only lasted as long as the previous sets of drugs. In my case, about 3 years. But these seem to be, from my evidence, and the big studies are yet to come out, really. Next lot will be in December, I think, at ASH. but they are game changing as far as I can see for a still as yet incurable blood cancer. More patients using them, survival rates are likely to increase, but the treatments will last longer and longer. So all these issues I mentioned will get worse and worse. The CRS and viral issues, obviously a lot worse for CAR T-cell therapy. And I can't speak to that because I haven't been through that, but you can see from the stats that Martin presented, that is definitely the case. They put a massive additional strain on hospitals and the health service. And I know through my work with Myeloma UK and the advocacy panel about how hard they work to get these drugs approved. The CRS and the other side effects are foremost in the minds of drug regulators, along with the prices, of course, and therefore of massive concern to the drug manufacturers. And of course, they impact the patient's quality of life massively. It's all very well not having the myeloma inside you, having lower levels, but you've got no quality of life. What's the point, really? So an efficacious oral prophylactic for or antidate to, as I said, oral being the key thing, CRS would help get patients out of hospital more quickly and reduce that strain. Interesting for hospitals, health services, drug regulators, and big pharma, and improve the patient's quality of life. And I'm all for that. Thank you very much. Thanks for listening. Hope that helps.
Brilliant. Bob, thank you so much for bringing us through your story, your 12-year story, I think with honesty, humility, with humor. I think it's good to see that, and delighted you can make it this far. I'm kind of envious of the cycling expeditions you've been on. I'll have to get out of the office sometime. But listen, thank you for your story, and I think you're highlighting again for us the importance of what we do. We do what we feel to be important work, but the end goal is to impact patients such as yourself. So again, appreciate taking your time to bring us through that again, give us that real world experience. Our final speaker before I come back in to wrap up, Professor Brendan Buckley. I know some of you, Brendan has presented before on a few of our other programs. Again, very important component to Pool, bringing lots of wisdom and sage advice. Brendan, isn't that right? But again, Brendan brings his medical and scientific expertise to POOLBEG, and he'll talk us through now, say, 001 as a potential treatment to manage CRS. So Brendan, over to you.
Thank you very much, Jeremy. Just to introduce myself very briefly, I'm a former academic physician, spent most of my life in hospitals and teaching, and came over to the dark side in industry a number of years ago to become the Chief Medical Officer of ICON, which is now the biggest CRO in the world, probably. And I also have a background as a member of the board of the national regulator here and involved heavily in the European Medicines Agency. I'm a co-founder, member of the board of directors of POOLBEG and on the scientific advisory board. I have to rely on my betters in the likes of Martin in hematology. I'm not a hematologist, but this is an area that has fascinated me over the last number of years. I must say that I thought, Bob, you were very inspiring, and it's very important that we're reminded by people like you why we do this kind of stuff, and that we need to keep our nose to the grindstone in developing treatments which will make life better for the likes of you and other patients as we go along. The thing that I want to address here is whether in POLB 001 we have something that is worth proceeding with and that has a decent hope of turning into a product which will tick the boxes that both Martin and Bob, from their different perspectives, want. And we certainly have an indication that this may be a worthwhile drug as it develops through. There are, of course, always risks, and we will only know at the end whether a regulator will approve this and whether payers will reimburse it. But at present, what we have is an oral drug. It's a small molecule. It's not going to be a complete breaker of the bank. as antibodies would be. It's twice daily dosing currently. It's very bioavailable, and really attractively, we know a lot about it, particularly in relation to its safety. This drug was developed originally by others from whom we acquired it as a treatment for rheumatoid arthritis. Again, because of its mechanism of action, cooling down inflammation in people with rheumatoid disease. And in the course of that, it was developing reasonably okay, except for an issue, which was that over the course of, say, a 6-month timescale, it caused early indications that it might be straining the liver a little bit. But to emphasize that this signal of safety took about 6 months to emerge. So in the short-term use that we envisage it here, this isn't a problem. It didn't proceed further with rheumatoid disease because it was overtaken by a whole new class of treatments in rheumatoid called the TNF-alpha inhibitors. So these just took off and everything was dropped in their wake. And POLB 001 was a fallout of those decisions of its original developers. So how does it work? Well, there's a pivotal point in inflammation, a kind of a nexus called MAP kinase, P38 MAP kinase. MAP kinases are proteins, enzymes inside cells that are turned on by a signal landing on the cell surface. So if you can imagine that from the outside, a signal comes to a cell which says, look out, problems are beginning to come. There's a problem on the horizon. MAP kinase is then switched on, and there's a cascade of signals passed through the cell to the nucleus. And the nucleus changes how the cell behaves. It might divide, or it may take various other actions to respond to a noxious stimulus coming by. The kinds of noxious stimulus might be the onset of a severe infection, or indeed T cells coming in and attacking a tumor. All of these activities send a ripple through the body saying that we're now moving out of a healthy state. The body is being, so to speak, attacked. We're going to have to react. And that reaction takes the form of inflammation. The molecules which tell the body that this is happening are called cytokines. There are many of these, and they generally act on a whole pile of different immune cells, different classes, as well as different numbers. So POLB 001 is able to inhibit the signaling which is telling the cell that it needs to be particularly distressed. And that cools down the panic which may arise when that signaling is too vigorous. So when inflammation comes into the healthy body, it may be appropriately dealt with by cells, but in some circumstances, the body may badly overreact, produce too many cytokines, and then you have a storm of inflammation, which we call cytokine release syndrome. And that can lead to a lot of damage as an unintended effect of inflammation. there are quite a number of these cytokines. In fact, there are probably hundreds of these described, which play a role in initiating and driving inflammation when it goes out of hand, as in CRS. And we have a variety of these things, various interleukins and TNF-alpha and so forth. And the more that cytokines are released, the more severe the CRS becomes. So these are a series of individual cytokines, and as their concentration increases, as they're released more by cells, CRS becomes more severe and the patient becomes more ill. So we designed a study to see whether we could calm down in a highly controlled experimental system, calm down cytokine release syndrome. And we did that in a so-called challenge study. We recruited healthy volunteers with no history of inflammatory disease, not on any medicines or any other inflammatory complications. And they were injected with a thing called lipopolysaccharide. This is sometimes known as bacterial endotoxin. This is a chemical substance comprised of lipid and carbohydrate together, which trigger inflammation in the body predictably. We gave this in 2 ways in the same volunteers. Initially, we gave them a dose of lipopolysaccharide under their skin. And then subsequently, we gave them an intravenous dose. These doses were done at a level which wasn't going to make the volunteers very sick. This is a well-established model, has been used for many years in exploring treatments for inflammation. The volunteers who were getting lipopolysaccharide were treated with one of a number of doses of POLB 001 in advance, or they were given placebo. And we looked at the response to the lipopolysaccharide injections, both in terms of the physical effects on them, looking at the effect on the skin through a little punch biopsy of a couple of millimeters of skin, taking their bloods to look for cytokines, and looking to see how they tolerated it, whether they got fever, what their ECG did, all that kind of stuff. And this is how the results looked like. First thing was that we were able effectively to knock down the P38 MAP kinase. So we were able to basically switch down this key triggering central component of inflammation. We can know that because we can measure the amount by which the proteins which are activated by P38 MAP kinase, we were able to measure how much they were activated because phosphate is stuck onto them in the process of the enzyme. So we were able to inhibit this phosphorylation of those proteins. And that was achieved in a dose-dependent form. So up here, this is the amount of phosphorylation here, where people got placebo. And then as they got increasing doses of the POLB 001, we were able to more or less completely knock down the activation of this enzyme so that no cytokine activity was triggered by it. Measuring a whole series of cytokines, and the big ones are TNF-alpha, interleukin 6, and interleukin 8. again, you'll see the same dose response curve of these cytokines. So placebo, they were at a high level. Small dose of POLB, they were cut down to about 2/3, and then they were almost completely knocked down. Not totally eliminated, but almost completely eliminated by our drug. To validate the clinical effects of this, we were able to see as well that POLB-001 in increasing doses blunted the inflammatory effect of increase in body temperature, blunted the rise in heart rate, and blunted the production of a protein which is indicative of overall inflammation called C-reactive protein. So this is an entirely coherent demonstration of the ability of POLB-001 to substantially cut down, to blunt the inflammatory response to the stimulus of lipopolysaccharide that we gave these volunteers. Now, if we can, in patients who get CAR-T or bispecific antibody treatments, if we can demonstrate that further, if we can validate this, not just in volunteers, but in patients, and seeing the effects in fever, heart rate, seeing the effects on cytokines, we should be able to prevent acute kidney injury and all of the other effects, respiratory failure, that end people with severe CRS inside in the intensive care unit, or even with grade 1 CRS, require them to be kept in hospital for days for observation to make sure that it doesn't progress to a catastrophe. In terms of the prospects for this, and again, trying to put my former physician's hat on me, a limitation to the availability of all of these new treatments, like bispecific antibodies, CAR-T, and so forth, is whether the health services can afford the drugs, but also can afford to deal with the consequences in the first few weeks after their treatment because of the risk and the actuality of CRS. And the mission for us is not just to help patients who have these treatments already, but it's also to expand the availability of bispecific antibodies and CAR-T therapies and others in the future, so that the huge promise of these wonderful new treatments isn't taken away from them by the practicality of how these patients are managed after treatment. What do we have at the moment for dealing with CRS? Well, we have dexamethasone. This is a kind of big, heavy gun steroid that's given intravenously generally, and that's been around for a long time. The other treatments, Actemra, tocilizumab, it's an old, well, it's 15 years old now, I guess, a rheumatoid arthritis drug, and anakinra. These drugs are not approved currently for the treatment of CRS with bispecific antibody therapy. So when they're used in clinical practice to treat CRS, they're used informally and off-label on the responsibility of the treating physician. So there are a number of other treatments that are being investigated for immunotherapy-induced CRS, but let's see how they work out. They, again, and in general, are likely to be quite expensive antibody-type therapies. While CRS is a problem now which isn't overwhelming hospitals, there are lots of new treatments in CAR-T and bispecific antibody therapies currently in development for, there are tri-specific antibodies that are in development. There are continuous improvements of these therapies to capture T cells and to get them to attack tumors. So the need for the effect of dealing with CRS is going to grow and grow with time. So there is no doubt whatsoever that there's a real need to advance POLB in oncology. And my belief from the data that I presented you in volunteer studies is that this small molecule treatment, about which we know a lot in terms of its safety, is highly promising in terms of its ability to cut down on potential cytokine storm, cytokine release syndrome with these therapies, so that they're enabled to be provided to people, maybe even on a simpler basis and in the fullness of time, potentially transforming them from medium-length hospital therapy into short-term hospital or even to an outpatient procedure with appropriate monitoring. Over to you, Jeremy.
Brilliant. Brendan, thank you for that comprehensive overview. I think you get the sense, everybody, for us here at Poolbeg, we feel we have a very exciting molecule, I guess very exciting programs. We can see clear benefits that POLB 001 can bring. I think with the story today, obviously we're kind of progressing in that direction. So, very happy with the direction that the company is in, and obviously, on the back of the exciting LPS challenge data we got this year, there's certainly a clearer path forward for Poolbeg in that regard. So what I'll do, just one last slide to wrap up before we go to some Q&A. But in conclusion, as I said, we believe 001 can address significant unmet medical needs associated with CRS across multiple settings. And I think that's a key aspect. We've always said in the past that from a pharma partnering standpoint, that having one molecule that treats different diseases is of interest to pharma. And I think when you're impacting or adjusting even the immune system, there's a lot of scope there for 001 to have a significant role. For cancer immunotherapy, as I mentioned, we filed patents earlier this year. We saw the potential there, and happily to see the significant interest of testing the impact that 001 can have on CRS. So as Brendan said, there's many of these immunotherapies coming through. And I think we can play a role as an adjunct alongside these treatments going forward. So again, CRS is a significant increase in the healthcare burden. I think Martin outlined it well with the various degrees of CRS, the impact that has on patient stays. Again, you're seeing the numbers from Bob and Brendan mentioned as well. If we can get patients out of hospital quicker, or indeed not in hospital at all, I think that allows more patients to come through. That allows less burden on the healthcare system, and it'll drive, as I say, patient responses will drive revenues for pharma. So there's multiple benefits on several levels here. So again, I think we're in a very nice position with 001, having the clinical data we have, that unlocking benefits of these efficacious drugs is further, where if you can remove the side effects, then the efficacy will be much cleaner and I think much better appreciated. So again, pharma companies will look to this and say this will drive their sales. They've got a good product, they've got good efficacy, remove the safety, and sales will increase and improve. And as Bob said, about the fact that he felt he was taking up a bed, I think that's obviously you trust your docs, etc. But he's right in the sense that if you were out of the hospital, there's more patients would be coming through, more patients would benefit in the long run. So again, our discussions with pharma partners have been very welcome and very positive. We've got colleagues at Bio Europe this week continuing those discussions. So again, as I say, there's multiple ways we can structure deals, getting pharma companies providing their drugs, of course, but then beyond that, when it comes to deal making and upfront payments and downstream milestones, etc. There's many different options we can take there. So looking forward to those discussions and negotiations progressing. So I think obviously it's key for Poolbeg. This is our prized asset. As I said, we've got a robust pipeline. But 001 is a prized asset, so we make sure we get the best deal possible for Poolbeg, and of course that'll have the knock-on benefits for shareholders, of course. So again, ultimately we'll see huge benefits for patients and the healthcare system as we, as I say, progress 001 into the clinic. Testing its potential to reduce CRS in these immunotherapy settings. So again, I want to thank you all for joining us. We've ran slightly over time, but we had a buffer because now we'll transition over to some Q&As, and so I'll kind of chair the Q&A session. I see there's a few questions in for Martin, etc. So we'll kind of go through these. I'll take the first question, pre-submitted. When can we expect to see commercial partner deal for 001 to see momentum in the lagging share price to at least above the spin-out price of 10p? Very good question. I think obviously we don't talk about times and timing. It's these if you were to kind of put a line in the sand, I think that gives pharma a lot more leverage into slowing processes down to get deals done. But I do appreciate the share price. And again, these are challenging markets. I think we're all aware, certainly in the life science sector. But I will commit. I mean, where we see kind of multiple avenues here for 001 and getting a deal done or transactions done. Obviously, can't talk about timing on that. But I think with the the discussions we've had thus far and the interest as a potential treatment for CRS, I think there's multiple avenues you can take, and we can kind of move forward in that direction. Martin, a question for you. Do you think that bispecifics and other such cancer immunotherapies could become a more standard treatment for multiple myeloma and others? It's currently used as quite a late stage of treatment, but with better CRS treatment, could this change?
Oh, definitely. I mean, they're going to move into earlier lines and wider lines of therapy in more applications for sure. I think a better CRS treatment would facilitate that and speed that up even further. I think the need for more and better CRS management is going to be there anyway because of their efficacy. The trials for frontline treatment with these agents are ongoing already.
Brilliant. All right. One more for you, Martin. I see here. Are there more oncology treatments in development that could have the same issue? So I think you touched upon that, but maybe a bit more kind of detail if you could.
Yeah, I think the efficacy of these treatments, some 10 years ago, it was highly debated whether a disease like myeloma will ever respond to a monoclonal antibody to the extent that we have seen first with naked antibodies, but then actually particularly the single agent activity of the bispecifics has been so high. People didn't believe that, especially because myeloma is originally a very immune protected tumor. It's different to these immune hot tumors that respond to PD-1 therapy. And of course, the proof of principle in some sense that in such a disease a bispecific antibody works is already being translated now into bispecific antibodies being clearly active in solid oncology as well, in tumors where also probably maybe even it will be some tumors where PD-1 therapy does not have the traction because they are not really immune hot. And I was really just stunned, ESMO, of course, thanks to social media, is much more accessible, and to suddenly see these exciting efficacy data coming through for the solid oncology that we normally used to see in hematology. But then whenever the slides flipped to the side effect profile, they had CRS on there, which was actually quite interesting, as I could already sense that the solid oncology colleagues here at my hospital will be knocking at my door quite soon to ask how we are managing that in practice for our patients.
Very good, very good. Yeah, I mean, we've had other discussions, just as an aside, with other key opinion leaders, and I think that's there's excitement about the future, but obviously it's like this, pardon the pun, headache, because I know that's one of the side effects, but it's this headache of CRS, and it's coming through, and as I say, it's just the burden on the healthcare system where people are worried about. The drugs are working, but can you manage this? And obviously we see potential here for Poolbeg. You can take a little break now, Martin. Bob, a question for you, if you don't mind. Before you started bispecific treatment, what was your biggest worry about starting?
Okay, obviously that's a loaded question. And it wasn't CRS, because my biggest concern was it going to work?
Yeah, of course.
And because I'm such a nerd, I'd done so much research on the studies that were out already, and spoken to lots of other patients, and spoken to lots of leading consultants. So my concern, as I said, was whether it would work or not. But having said that, I speak to a lot of other patients. A lot of people have seen my story on various forums and have contacted me, and that is the first question they ask me: what about the CRS? And I understand that, because as I said, I looked at CAR T-cell therapy in the last 3 or 4 years. I've been following the progress, thinking this is going to be the breakthrough for me. But my wife and I watched a first-in-human program about CAR T-cell therapy in the States, and of the 3 subjects, 2 of them died. And so that's quite hard hitting. And other people have heard that, and that's what it gets a reputation for, potentially being fatal. And patients who on the anxious side will just focus on that. So hopefully I reassure them that that's usually the case. But anyway, that's really, I'm the wrong person to ask about worrying about things before they start, I'm afraid.
It's the right attitude, I guess, Bob. But I just think it's a good question, because you were, as you called yourself, like a geek or a nerd? You were calling yourself. I mean, you've done all the reading, and you knowing what to expect. But I think with these newer treatments, I can imagine there's a little bit of stress to see how it turns out. But thankfully, you're still going strong. That's good. A question. Let me see. A question for me, I guess, from Kevin H. Do you now see the potential for POLB 001 as greater in oncology, cytokine storm, than in influenza? That's a really good question. I'm guessing you or a partner can pursue 2 different indications with the same compound. It's a really good question. Today's focus obviously is oncology, but I will refer back to severe influenza. That is still an issue. It's I think 3 to 5 million people globally annually get severe influenza. There's 500,000 deaths, and we do see there's a potential there for that. I think with the oncology, and I say this is relatively recent in our discussions, there's really keen interest in in pursuing the oncology arm as well. So it's not one over the other as I see it. But the question about pursuing 2 different indications, it's an interesting one. It's certainly feasible, and we've talked about this in the past. I believe that if we're looking at somebody in the oncology space, that we talk about the CRS, we see this as it's a small molecule, so we see this as kind of an orally delivered drug. So it's it could be a small molecule, or a pill form given to the patients to manage CRS. For severe influenza, that's a different setting. That's more in the hospitalized ICU setting, where these people are obviously had the infection, the cytokine release syndrome, hypercytokemia is progressing. So these patients are more or less based in the intensive care unit, where it's more likely going to be an IV formulation, so intravenous rather than oral, because they may not be in a position. So there is ways to kind of carve up the world. You could do license deals with regard to just oncology, focus there on infectious diseases, focus there. In an ideal setting, obviously, it could be the same pharma company who pursue the direction in both. So obviously, we've got many possibilities that we're leaning towards there. So again, we'll keep the markets updated as we progress. Let me see the next one. Maybe this is back to you, Martin. I see some specific wording here, but what advantages do you believe POLB 001 or a P38 MAP kinase inhibition can offer over dexamethasone or other drugs used for managing CRS, such as siltiximab, tocilizumab, or anakinra? I may have some thoughts on that as well, but do you want to start, or will I start?
Yeah, I mean, I think that it's clearly from the early data that we've seen that you've shown. The fact alone for it being oral is of course a big advantage over the key drug that we use probably in our myeloma setting, most tocilizumab as the first instance. And if that was actually able to abrogate CRS in the same way, that alone opens up completely different healthcare utilization than an IV drug as we use tocilizumab would have.
Yeah.
Dexamethasone, of course, is in comparison a dead old drug, and it's kind of on the one hand side a very powerful drug and still amazing in terms of the chemistry work that was done at the time in terms of producing the synthetic steroids, but it does have in some patients really quite severe psychological or even psychiatric side effects. So I think it's even in everyday practice there's always challenges of using drugs like dexamethasone, especially because they're very potent and can really derail some patient's situation quite quickly.
Thank you, Martin. Thank you. Yeah, one additional thought we had was, particularly when we were comparing it to the tocilizumab, the anti-IL-6, that, and Brendan has shared this data, that not only are we hitting or reducing IL-6, but we're also seeing reductions in TNF-alpha, in IL-8. We saw CRS. So it's while our POLB 001 is specific and selective for P38 MAP kinase, it has an effect on lowering kind of more than just one cytokine. So we think there may be benefits, but of course the oral as well. That's it's an important one from a healthcare perspective. That question just to tag was from Robin D. So appreciate that. Sparky J has a question. Have you an interested party in oncology use for 001 who suggested that you explore that angle as it's something that they may be inclined to sign a partnership deal for, rather that use rather than RSV use, or I presume severe influenza? I mean, as a company where we're very much partnership or business development focused, we've talked to a lot of farmers. When we talked to them about CRS and oncology, they were really certainly interested. They saw the potential here. They saw the issues that they had with the data generated in clinical trials from their immuno-oncology drugs, the bispecific antibodies. Yes, I mean, we brought it to them and they see the benefits of that. So there is discussions going on about, as I say, running this trial. We talked about getting key early human clinical data. Again, it's a key tenet for the business strategy for POOLBEG. So there is interest in that. Now, I wouldn't say it's one over the other. It's just you talk, like, pharma companies are huge and large. So, it's not the same people you talk to when you're talking about infectious diseases versus oncology. But we certainly have strong interest on the oncology side. Let's see here. Brendan, maybe one for you. I see here from Emily L. Will POLB 001 need a specific regulatory approval for each cancer or treatment? In other words, will 001 need a regulatory approval for myeloma and another regulatory approval for small cell lung cancer, as an example?
I don't believe that will be the case, Emily. It's the indication here that we would be thinking of looking for would be for cytokine release syndrome, specifically of whatever cause, as long as it was caused by intervention in oncology treatment, for example.
Yeah.
So it's perfectly reasonable to have an indication which relates not to the very underlying disease, but to the condition itself. And regulators talk about conditions rather than diseases, and the condition here is CRS.
Yeah, very good. And I think, Brendan, you'll recall some of the discussions we had from a strategic standpoint. The fact we'll start obviously kind of with the myeloma side. That's where our kind of key opinion has brought us. But we do see potential way beyond that. CRS is an issue beyond just kind of myeloma in other areas.
Absolutely.
So we're obviously kind of thinking strategically there. So even though we get that single approval, the drug can be applied broadly, which I think is of great interest.
And to kind of go back on a little bit of the question about the influenza side of it, fundamentally, it's cytokine release syndrome of whatever cause.
Yeah.
And so what we learn in oncology, we can apply in infectious disease and vice versa.
True.
So because ultimately we're looking at the same cascade of the same kinds of molecule.
Great. No, really good point. Maybe I see the time is ticking rapidly by. Let's just do one more question. Again, Martin, if we could finish with you again from Robin D. One question for the clinician, perhaps. Does the risk of CRS currently influence the choice of anti-cancer therapy? That is, are there some patients who may be too fragile to survive CRS that may lead to a different therapy choice to one where there is a lower risk? Really good question.
I think at the moment we're at that stage where, these therapeutic options are still being kind of like in the phase of development, that we don't really have them as competing options yet as far. But actually, I can very easily see a future where that is the case. Definitely in our practice, for example, CAR T cells play a lower role, but they do play a higher role in other markets like the US.
Yeah, sure.
And there clearly you would definitely tune, maybe not only because of the CRS, but the whole package of the inpatient stay, the conditioning chemotherapy that the patient needs to receive, the infection risk afterwards as well. But CRS plays a role in there. And yes, you would choose certain treatments for example, for elderly or frailer patients over others that are more intensive. Now, having said that, however, even the less invasive therapies or less intensive therapies would still come with a CRS risk. So, for example, a bispecific antibody we have found, are really very deliverable in an elderly population, although, of course, more and more data will be required to back that up. But I think both here, as well as particularly in the US, where these drugs have been licensed for a longer time, there's already a large amount of real-world data amassing in that space, because the unmet need is clearly there anyway to treat these patients.
We're always learning, Martin. We're always learning. Super. Listen, I see the time. Let's wrap it up there. And can I just extend my my grateful and thanks to Martin, to Bob, to Brendan for joining us today. Going through, as I say, this is an exciting direction that Poolbeg is taking in the oncology sector. So we just wanted to take the last 90 minutes to give the audience an update on that direction. I hope it was informative. I hope it was useful. And I think it gives you that kind of real world approach of what we're doing. So again, thank you everybody for your time, and thank you again for our guests here today. Let's call you guests for joining us in the discussion. And we look forward to keeping people updated on Poolbeg's progress in the near future. Thanks, everybody. Bye-bye.